Hi, DrBiology here, and this video is about inherited diseases, and it's part of the topic of inheritance, variation and evolution for AQA, GCSE biology and combined science. So this is part 4 of a series of videos on this topic on inheritance, variation and evolution, and we're going to look at inherited disorders in this section. So sometimes genes do mutate, and that's a mutation in the genetic code, and therefore causing which proteins can be coded for, and causing a change in the genetic code. Some can be passed from parent to child, therefore they are inherited, but some cause few problems, so things like colour blindness, for example, but others can be fatal. Now, for your specification with AQA, you need to know specifically about 2 genetic disorders, which is cystic fibrosis and polydactyly. So cystic fibrosis, so the exam will expect you to know what is it and how is it treated, and also then about the genetics behind it. So it is an inherited disorder of cell membranes that mainly affects the lungs and digestive system, so they become clogged with thick, sticky mucus. And over many years, the lungs can be increasingly damaged by infections, for example, and other problems, so they eventually can stop working. It's mainly treated with physiotherapy and antibiotics to help the lungs keep clear of mucus. Enzymes are used to replace the ones that the pancreas cannot produce, and also to thin the mucus. And there is still no cure for this disorder. It's caused by a recessive allele. So a child has to inherit 2 copies of this faulty gene from their parents, one from their mother and one from their father. So in this case, capital F is the dominant allele and the recessive allele is shown as a lowercase f. So if the parents have a heterozygous dominant, in other words, big F, little f, they are known as carriers. Now, the parents won't have the symptoms of cystic fibrosis, but they carry one faulty gene or one faulty allele. So if you inherit both recessive alleles, then you have cystic fibrosis. As you can see, if your mother and father are carriers, there's a 1 in 4 or 25% chance that any offspring that they will have cystic fibrosis. And conversely, there's a 1 in 2 chance that they will be carriers, and a 1 in 4 chance that they will not be carriers and not have cystic fibrosis. Polydactyly is an inherited disorder in which a person has extra fingers or toes. So sometimes the extra digits can be removed by surgery, usually at a young age, or if they don't cause a medical issue, they will be left. It's caused by a dominant allele of a mutated gene. So this means it can be passed on by just one allele from one parent if they have the disorder. So in this example, in this Punnett square example, you can see the mother is unaffected, but the father has the condition because they have one dominant allele. So you can see that there's a 50% chance of having the disorder and 50% chance are unaffected. Now, sometimes in the exam, they will show you things like this, which is a family tree. So these can trace genetic characteristics through a family tree. And you can see from here that the initial parents, John and Susan, so Susan is a carrier, and you can see their offspring, so Mark, Caroline and Phil. Caroline is unaffected, whereas Mark and Phil are carriers. Now, then you can see that Phil gets together with Eve, and they're both carriers. So the question is, what's the chances of their new baby having cystic fibrosis? Okay, so as we said, the allele for cystic fibrosis isn't dominant, as many of the offspring are carriers. So you need the recessive allele, one recessive allele, to be the carrier. But even Phil are carriers, so the new baby has a 1 in 4 or 25% chance of any of their offspring having cystic fibrosis. So the new baby has a 25% chance of having cystic fibrosis. So you can see how they're useful for predicting genetic characteristics. Not just genetic disorders, but many different types of characteristics. Okay, so genetic testing. So genetic testing involves the analysis of a person's DNA to see if they carry alleles that cause genetic disorders. And it can be done at any stage in a person's life, but usually it's done at an early stage in the pregnancy. Now, one of my top tips here is that in the exam, you will be given plenty of information to analyse and evaluate. So you wouldn't be expected to remember everything that is in these diagrams. You might be expected to know some of the facts that I'm giving you, but I will be showing you later some exam questions where you need to analyse and evaluate information. So the first type of procedure is amniocentesis. So this happens at 15 to 16 weeks of pregnancy. Fluid is removed from around the developing fetus, and these contain cells that are used for genetic screening. So what they'll be doing is looking for a genetic disorder, and then deciding whether the baby needs to go to full term or not. One of the main risks of this is miscarriage, so in the first 23 weeks of the pregnancy. So this is estimated to occur in 0.5 to 1% of women who have amniocentesis. The second stage is chorionic villus sampling. Luckily, you don't need to remember the full word for that. If you just remember CVS, that is completely fine. So it's at 10 to 12 weeks of pregnancy, a bit earlier, and you can take samples of embryonic cells from the placenta. And again, the risks are miscarriage and also the same kind of risks of having CVS as in amniocentesis. Okay, so embryos produced by IVF. So some couples will go for in vitro fertilization. So this is where an egg is removed from the mother and then sperm is added in a petri dish, and then embryos are produced outside the mother. And these are tested before they're implanted back into the mother. So the advantage of that is there's no risk of miscarriage as a healthy embryo is implanted. There are many arguments for and against these procedures, and in your exam you will be given information to evaluate. Now, it might be worthwhile learning some arguments for and against genetic testing that will help you, but obviously if they say use the information, then you can use that information and maybe add some of your own information. So arguments for genetic testing are that it will help stop people suffering from genetic disorders. Also, treating disorders does cost the government a lot of money, and there are laws to protect unborn babies. Now, the arguments against are that collecting cells from a fetus can cause a miscarriage. Screening is very expensive. It does imply that people with genetic problems are undesirable, and it could also give rise to people having too much choice. In other words, something called designer babies. So choosing not just those babies that don't have disorders, but babies with certain characteristics. And that's not really seen as a positive thing in society. Okay, so let's have a look at some questions. So this is a question checkpoint, and what I'm going to ask you to do in a minute is to pause and read and try and answer the question. Okay, so you can pause in 3, 2, 1. Okay, so this first question is for foundations here, and you can see that it says draw a ring around the correct answer to complete each sentence, and it asks you the allele for cystic fibrosis. So this will be from your knowledge, and the answer is recessive allele. Mr. and Mrs. Brown are both what? Well, they don't have cystic fibrosis, but they have a child with cystic fibrosis, so they must be carriers. Then it shows how the diagram shows how the allele for cystic fibrosis is inherited by Mr. and Mrs. Brown's children. It says, give the number of one person in the diagram who has cystic fibrosis. So you can see that the two recessive alleles are little n and little n, so that must be number 6. The chance that Mr. and Mrs. Brown's next child will have cystic fibrosis, well, it's the same as their first child, so it will be 1 in 4, or you could have 25% or 0.25. Second part of the question, again, this is a foundation tier, but also might be used in higher tier. They give you lots of information about genetic counselling, and they show you some what they do in terms of testing the embryo. And it says, the question says, use the information to suggest one advantage and one disadvantage of screening embryos in this way. So I'd like you to read it, I'd like you to pause and then try and answer. So 3, 2, 1. So looking at the information, the advantages are that they can detect cystic fibrosis at an early stage. A disadvantage is that some embryos may be destroyed or damaged, or it's going to increase the risk of miscarriage. Question 2 is about polydactyly, and it tells you that it's caused by a dominant allele, and it shows a man with polydactyly and he has an extra finger on each hand. The man's mother also has polydactyly, but his father does not. So I'd like you to read the question and pause and have a go at it. Okay, so 3, 2, 1. So the first question, the man is heterozygous, and it wants you to explain how the information given above shows that the man is heterozygous to polydactyly. Well, the fact is it's 3 marks, so you've got to say that firstly, the man inherited the PD allele from the mother. The man has normal recessive allele from the father because the father doesn't have it. Okay, so the father does not have the PD allele, so that's why it's heterozygous. And then it says the man marries a woman who does not have polydactyly. What is the probability that their first child will have polydactyly? Well, you could draw out a Punnett square diagram at this stage, okay, but usually, all right, when they don't have it or they do have it, it's always a 50% chance. So you can have either 0.5 or 1 in 2 or 1 to 1 or 50%. So this question is definitely a higher tier question, and it's a 15-mark question as well. So we'll go through it together. So first of all, I'd like you to have a look at this question, and it talks about CRAM. So CRAM is an inherited condition. Now, as you've noticed, I haven't told you anything about this inherited condition. So sometimes they may give you conditions you've never heard of, and you need to look at the information very carefully. And the key bit of information there is that CRAM is caused by a recessive allele, lowercase n, and the allele for normal health is capital N. So have a look at it, and I'd like you to try and answer those questions. So pause in 3, 2, 1. So let's look at the questions. So the allele, question A, sorry, what is an allele? So straightforward, it just wants some information on recall. So it's a different form of the same gene or of a gene. What does recessive mean? Well, it's not expressed if a dominant allele is present, or you could say it will be expressed if both alleles are recessive, homozygous, for example. Give evidence from the diagram that CRAM is caused by a recessive allele. So you need to look at the diagram and you need to decide which ones. And you'll notice that unaffected parents 7 and 8, they have a man with CRAM. So they have an affected child, number 10. So therefore, that is saying it's caused by a recessive allele because the parents must be heterozygous dominant, and the child, number 10, has inherited both recessive alleles. Person 2 is a homozygous for CRAM. What does homozygous mean? Well, homozygous means that the 2 alleles are the same. So let's look at the second part of the question. So again, I'd like you to have a really good look at this. I'm going to tell you that C, you will have to draw a Punnett square diagram. So it says draw a genetic diagram to explain your answer. So pause in 3, 2, 1. So it says none of the person's 2's children have CRAM. Explain why. Well, it's because they've inherited the dominant N. So all are heterozygous dominant. Person 7 and 8 want to have another child. So if you remember, if I just show you, I'll just go back they have normal health, they have recessive, they have heterozygous dominant, so they have a dominant and a recessive allele. Okay, so what is the probability that this child will have CRAM? So draw a genetic diagram. So here we go. So I've drawn the genetic diagram, so you can see the parents there and you can see the offspring, and the probability is 0.25, and I'll show you why. Because basically this is the person, so it wants the probability. So probability is shown in decimals, which is 0.25. So you can't show it as a ratio, for example. Right, this question is a bit more involved, and they give, as I said, they give you lots of information about different types of genetic screening. All right, and then they say the question is evaluate the benefits of these two methods. All right, so you need to look at the benefits of both methods and then include a conclusion to your evaluation. So again, I'm going to ask you to pause and have a go. So pause in 3, 2, 1. Right, so it's quite an involved answer here. Okay, so pro PGD there's lots of answers here. You don't need all of these answers. So less chance of miscarriage, doesn't involve abortion, higher chance of unaffected children, provides embryos for research, so that's pre-implantation. CVS, PGD may destroy some embryos. Okay, so there are some problems of PGD. Pro CVS means that there's a lower incidence of false positives and basically a less financial cost, and then you've got to relate it to your argument. Okay, and it says you must have at least 1 point from each technique for maximum marks. Okay, so that brings us to the end of this video. I hope you found it useful. If you're not already, please do subscribe. Make sure your bell alert is on and notifying you of any new videos. I'll see you soon.